Thank you. So, hi everybody. So, yeah, I'm from the. Sorry, it's pointer. Pointer should be here. Oh, yeah. Okay, great. So I'm from Beijing Institute of Genomics, Chinese Academy of Sciences. So today I would like to share some of our recent work on the CIRI-Deep, which is a deep learning model of the circular analysis. Yeah, so we all know that so splicing is an important step in RNA processing. So it removes introns and joins exons to produce mature mRNAs. But there's also a special form of splicing, which removes all of the upstream and downstream sequences as introns and join the exons in the opposite direction compared to the canonical splicing, which we call the back splicing. And the backsplicing can generate RNA molecules in the shape of circle, which we call the circular RNAs. So previous studies have already demonstrated the diverse function of circular RNAs. For example, some of them can function as a sponge of microRNAs. So they can indirectly regulate the translation and degradation of mRNAs. And from more than 10 years ago, we began to develop the computational methods for the circular RNA. We have a tool series called CIRI. CIRI can do the de novo detection of a backspace junction, BSG, from the RNA-seq data. And so we know that even in human, there are a large proportion of circular RNA that are not annotated. And if you are doing research on some non-model organisms that don't have complete annotations, you can choose CIRI. And the previous benchmark studies have already demonstrated the robust performance of CIRI and its later version CIRI-2 on both the identification and quantification of circular RNAs. We also have this latest version, CIRI-3, for the analysis of large dataset at the cohort or TB scale. Basically, you can use your laptop to finish the detection of circular RNA in just a few hours for a cohort of 100 people. However, it should be noted that not all of the RNA-seq data are equally suitable for circular RNA analysis. So we all know that most of the RNA-seq data are derived from the polyselect library. And this type of data are suitable for the analysis of mRNA, but because the circular RNA don't have the poly tails, so so the reads from the BSG cannot be easily detected from the polyselected RNA-seq data. By contrast, the ribosome RNA depleted library are more suitable for the analysis of circular RNAs. We also call this type of data the total RNA-seq data. Furthermore, for the single cell and the spatial transcriptomics, almost all of the data are derived from polyselect data, and most of them, such as those from the 10x genomics, only sequence the 3' or 5' end of the RNA molecules, which makes them unsuitable for the analysis of circular RNAs. So to fill this gap, we developed this CIRI-Deep. So it's a deep learning model for the BSG read independent prediction of circular RNA regulation between the biological samples. So this model is trained on over 25 million high confidence differentially spliced or unchanged circular RNA events across nearly 400 human tissue samples. Okay, so in CIRI-Deep, we use both cis and trans features. For the cis feature, we collected over 3,500 genomics features that are different between circular RNAs. And so they include this transcript structure, RBP binding motif, and allele sequences, and other cis-acting elements related to the canonical and back splicing. For the trans feature, we incorporated a gene expression of about 1,500 RNA binding protein. So this RBP included the splicing factors and RNA degradation enzymes and other genes related to the biogenesis of circRNAs. So we actually trained two models. One of the models is called CIRI-Deep, and it accepts the RBP expression from total RNA data, and the other one is called CIRI-Deep-A, which accepts the RBP expression from the polyA selected data. So our model have pretty good performance. For example, in the leave-out data from samples that were never used for the model training, our model can achieve an AURC higher than 0.9. And so when the sequencing is not deep enough, we can use the CIRI-Deep model to identify these differentially spliced RNAs, the DSC, more effectively compared to the traditional, to conventional statistical model. And also, using this CIRI-Deep-A, we can predict the DSC that are highly consistent with those we identified in the total RNA-seq data. You know, this deep learning model are often criticized for being difficult to interpret, right? So to calculate the contribution of these two types of features to our model, we use this adapted integrated gradients, the IG approach. Basically, we just fix one type of features when assessing the other type of features. For example, so if we assess the contribution of trans features, we will just fix the circRNA. We keep the circRNA unchanged to fix their cis feature, and we will calculate the IG of RBP expression in the path from the samples without the DSC to the sample with the DSC. And interestingly, we found that the contribution of the RBP expression are quite different across different samples, across different tissues, which shed light on the tissue-specific expression of circRNAs. And so we can also use this CIRI-Deep-A model to predict the DSC between cell clusters. For example, we applied our model to the 10x genomics data, which successfully identified the cell type specific circRNA that are sporadically detected in this corresponding massive data. And this is the spatial transcriptomics of a human heart. We just predict the DSC between the spots dominated by the cells from ventricle, atrium, and outflow tracts. And the result is highly consistent with what we identified in the corresponding bulk data. And we also converted the DSC probability between one spot with all of the other spots to an index indicating the BSG ratio on the map of spatial transcriptomics. So as you can see here, so this circRNA has a higher BSG ratio in the region of outflow tract, and in contrast, this one has a high BSG ratio in the ventricle region. Okay, I would like to thank all of the collaborators and the foundation support the research of our lab. Thank you.
Okay, we have time for some questions.
Hi, great talk. I guess I have a question about like the data split part, because you mentioned that you're using the sample pair to like split the data. But if you use the sample pair, wouldn't one of the sample in that pair who actually appear in the training data, or would that affect the analysis, or would that be a little kind of like the data leakage to the from training to the test data set, or in this specific problem that wouldn't be the case?
Yeah. So yeah, that's a good question. So basically, we used nearly 400 samples But as you mentioned, if one of the sample is from these samples, the data set, so usually it will have a better performance, yeah. But our performance shows that even for the data that are not trained in our when we train the model, when we still so the generalization capability of our model is pretty good according to our evaluation. Is that your ask? Is that what you asked? Yeah. Okay. Thank you.
Okay, I'll ask one more question. I'm kind of curious, how might you be able to, I don't know whether or not this is even possible, to incorporate long read sequencing or anything like that to be able to get a full length circular isoform? I know there's some experimental tricks here, but I'm curious your thoughts.
Yeah. Actually, that's a very good question. So that's actually what we are doing now. So I did not mention it because I don't have enough time. But basically, when you want to predict the full length isoform, not only the circular but also the mRNA transcript, the long reads are always a better choice according to recent papers published. So yeah, I think that's totally possible, and that's what we are doing. That.
Great.
Thank you.
Okay, awesome. Let's thank Yuan again.